Is it recording now?
Yes, sir.
Okay. Then let us start from the most important uh, two alkaloids, vincristine and vinblastine, which are produced by uh, Catharanthus roseus. Catharanthus roseus, popularly in Bangladesh, it is called Noyantara, and Noyantara plant is a herb uh, available and abundant in Bangladesh, and this is a very tolerant, stress tolerant plant, and this plant we use as an ornamental plant. Uh, as I mentioned, this plant produces two very important alkaloids. One is vincristine, another vinblastine. And these two alkaloids, why they are important? Because they are important biologically active compounds against cancer. Cancer, you know, one of the deadliest disease. Here you can see an image of how cancer cells are damaged by the uh, Noyantara alkaloids, that is vincristine and vinblastine. These two compounds are commercially very famous as anti-cancer drug. Here we shall discuss a little bit about vinblastine and vincristine. Uh, uh, they are also called vicindole type alkaloids. And if you go Wikipedia link about vincristine and vinblastine, you can get more in-depth discussion about these two compounds. Vinblastine is a vinca alkaloid. It is also called vinca alkaloid. It starts from the prefix vin and a chemical analog of vincristine. That is analog. You see both structure are same. One is CH3, another is CHO in a certain structural part. All other parts are same. This is why they are analog to each other. And how these two compounds, work against the cancer cell. The vinblastine binds tubulin. Tubulin is a protein. Protein means cytoskeletal protein. Cell has a cytoskeleton, and cell cytoskeleton composed of two types of protein. One is tubulin, another is actin. So it binds with cell cytoskeleton tubulin, vinblastine, and thereby inhibiting the assembly of microtubules. Tubulin is composed of microtubules. So assembly of microtubules actually result the main structure of the tubulin being polymerized protein. And it is a G1S phase. In cell cycle, you know, G1S phase is very important. Cell cycle specific G1S phase, this blastine actually work and inhibit the assembly of the microtubules. Vinblastine is a component of a number of chemotherapy regimens, including ABVD for Hodgkin's lymphoma, and it is also used to treat histiocytosis and many other cancerous cells it can cure. Vincristine, another compound, vincristine, this one, which has extension, structural difference only CHO connected to this N. So vincristine binds to tubulin, also binds with tubulin dimers, causing disassembly of microtubules. The first one inhibit the assembly. Assembly means microtubules assemble and produce the tubulin. And here, tubulin is a dynamic protein. Assembly and disassembly is taken place in the cell. In case of vincristine, it interferes the disassembly tubulin dimers, causing disassembly of the tubule structures. Assembled, tubulins become disassembled in presence of in crystine, and microtubules is very critical and very important in cell division. So if you can target any compound targets the microtubules or actin, the cell division can be stopped. And in case of cancer cell, the real problem is the cell always know the cancer cell knows only to divide and never goes for apoptosis. Apoptosis means the programmed cell death. In our body, all the cells has certain life cycle. After that, they die. But in case of cancer cell, never dies. Only divide, and become a tumor, and especially in the meta phase stage, vincristine does its function. The vinca alkaloids therefore affect all rapidly dividing cell types, including cancer cells, but also as intestinal epithelium and bone marrows. Main uses are in non-Hodgkin's lymphoma as part of the chemotherapy regimen CHOP, and Hodgkin's lymphoma as part of the Stanford V chemotherapy regimen. There are different chemotherapy process to treat the cancer patient, and these are two types I mentioned. And in acute lymphoblastic leukemia, that means vinblastine can be used against the leukemia cancer. The market price for vinblastine and vincristine extracted from plant is estimated at 2 million per kg. That means 1 kilogram of this compound is 2 million US dollar, and total 2 million US dollar vinblastine, and vincristine 15 million per kg US dollar. 15 million US dollar per kg. So vincristine is more expensive, 7.5 times more expensive than the vinblastine, and they are very powerful anti-cancer compound. So you may be asked, what are the structural differences between vincristine and vinblastine, which contain in the extension the radical CHO, which contains CH3. It may be asked you the question, how these two compounds do the function against the cancer cells. So the mode of action, the mechanism of action of these two compounds described here, you need to learn thoroughly. And these two compounds most famous anti-cancer drugs. And trafficking indole alkaloids metabolism already we discussed. Although they produce in the outer cells, they finally move to the vascular system and can move to one part to another part. For example, nicotine is biosynthesized in the root, and they are deposited in the leaves. Streptocidin derived quinolin alkaloid. This is another very important, historically very important alkaloids discovered from the cinchona bark. Cinchona plant produce quinine type alkaloids, and quinine, here you can see the global map. Malaria is a very very dangerous disease in tropical and subtropical regions, transmitted by the mosquitoes, and malaria is caused by the plasmodium falciparum, their parasite. So malarial parasite to kill or to cure the malaria disease, quinine have been used as a popular drug for a long time. Recently, quinine resistant Plasmodium falciparum have already been identified. So quinine discovered from cinchona bark is also very important important, how to say, therapeutic drug against the malaria. Properties of quinine. Quinine is present in the bark of various trees of the genus Cinchona. Cinchona is the genus and family is Rubiaceae, which are native to tropical South America. Uh, and these quinines are antipyretic, anti-malarial, with analgesic and anti-inflammatory properties. They have two major properties quinine has. One is analgesic, another is anti-inflammatory. This is why when quinine is used, malarial disease, when malaria attack human suffers from huge, pain, body pain, and headache, and parasite also causes anti-inflammatory problems. So anti-inflammatory principle of quinine can cure the malarial disease, and it is also followed by the synthetic analog of chloroquine. You have recently heard about chloroquine and hydroxychloroquine. The chloroquine and hydroxychloroquine, they have been discussed a lot as treating agents for the COVID-19. But there are huge controversy. Recently, one paper is retracted from Nature that claimed that chloroquine and hydroxychloroquine are effective drug against the COVID-19. They have huge side effects. So higher doses of chloroquine and hydroxychloroquine, that means this quinine, are problematic. And quinine is natural product, but chloroquine is a synthetic analog. That means structurally similar, but not the natural one. Now again, the drug of choice for treatment of Plasmodium falciparum malaria, because of the rise of serious problem in our region. Now it is a big problem in some African countries. Quinine interferes with the parasite's ability to break down and digest hemoglobin. You see, malarial parasites targets the hemoglobin because hemoglobin is needed for completing their life cycle. So uh, the quinine interferes with the parasite's ability to break down and digest the hemoglobin, thus starving the parasite. So parasite starved, and then parasites die. So this is very important mechanism. So if you are asked how the quinine works on the Plasmodium falciparum, here it is written very clearly that quinine interferes the parasite's ability to break down and digest hemoglobin, thus starving the parasites and or causing the buildup of toxic levels of partially degraded hemoglobin in the parasites, and then parasites cannot multiply, and a parasite cannot function against the uh, you know, uh, uh, human. Non-medical use in popular drinks. Quinine sometimes used in the tonic. That means different bitter lemon or tonic water. So a small quantity is low doses of, I mentioned, dose is most important and critical for toxicity of any compound. Although quinine is a toxic alkaloid, but low concentration people use in different drinks as they have some physiological effect in human body. Proposed pathway for the biosynthesis of quinine. Quinine biosynthesis is very complex. Here you can see how stick to acidine converted into the quinine, cinchonidine and quinine. Cinchonidine, quinine are analogs, and they are produced naturally from the cinchona plant. Another important anti-cancer drug is camptothecin. Camptothecin has been identified in phylogenetically unrelated plant species. Most of the secondary metabolites are have the chemotaxonomic relationship. But in case of camptothecin, has been identified phylogenetically unrelated plant species. It has anti-tumor activity. It is also one of the anti-cancer drug. And it is activated by hydrolysis of SN38, an inhibitor of topoisomerase 1. So it is an inhibitor of topoisomerase 1 enzyme, and it causes hydrolysis to the SN38. This is then inactivated by glucuronidation by uridine diphosphate glucuronosyl transferase enzyme. Eventually, this process inhibits DNA replication and transcription. That means camptothecin somehow inhibit DNA replication and transcription of the cell. So if you remember, this part is very important and critical for your exam. And this is commercially being available as a chemotherapeutic agent, and total market is 1 billion US dollar per per year. We are now starting to discuss another interesting alkaloid. Can you guess it? Purine alkaloids. Purine alkaloids, caffeine, especially a very popular, and this alkaloid found in coffee and very good for human health. Caffeine has antioxidant activity. This is why it boosts up the immunity, and regular 1 cup of coffee is good for human health, and it is considered as one of the best drink in this world. Uh, uh, there are several other alkaloids. Betalains. Betalains are a group of water-soluble chromoalkaloids, colored. You can see it found in the beet, uh, uh, red, violet, beta-cyanins, and yellow beta-xanthines, which occur only in certain members of the plant order Caryophyllales. Red beet, people use as vegetable because it contains very high concentration of betalains alkaloids, and very good. They are powerful antioxidant and good for human health. And some an anti-magic herb for centuries. This is very interesting. These plants is called anti-magic herb, and most interestingly, it was considered a reliable defense against witches. I am not sure, but people believe it. Artists, including Michelangelo, very famous artist in Europe, and Michelangelo, he was the artist of, uh, you know, Sistina Chapel Museum in Vatican City. The whole roof of the Vatican City church is full of art of Michelangelo. The story is according to the Bible, how human was created and all other things. And Leonardo da Vinci, another great artist. Both Michelangelo and Leonardo da Vinci improved their creativity and eyesight by ingesting rue. So ingestion of rue, eating of rue helped them to improve their creativity and eyesight, it is believed. Rue is a symbol of regret, sorrow, and repentance, and Catholics used springs of it to sprinkle holy water on worshippers. The rue leaf, you see, a card, playing card like clubs. The leaf is like that, and they produced a different kind of acridone alkaloids which help human creativity as well as eyesight as well as focus something very strongly. Uh, uh, but rue was formerly used to treat almost every ailment known to man, but its effectiveness and safety are questionable. You know, in case of herbal medicine, some of the activities are not scientifically proved, but people traditionally being used. This is most interesting. Another alkaloid, colchicine. Perhaps you heard about it. This colchicine. Colchicine is a powerful, cytotoxic alkaloid extracted from the plants of genus Colchicum, originally used to treat rheumatic complaints and especially gout. It was also prescribed for its cathartic and emetic effects. Colchicine inhibits microtubule polymerization. This is the most important principle of colchicine. Microtubule, as I mentioned before, the cell cytoskeleton requires polymerization during the cell division, and colchicine inhibits it. But colchicine do another very interesting by binding to tubulin. Microtubules cannot bind to tubulin. Colchicine inhibits microtubules. Apart from inhibiting mitosis, a process heavily dependent on cytoskeletal changes, it also inhibits neutrophils' motility activity, leading to a net anti-inflammatory effect. So it has some medicinal principle, but the problem is it directly affect the microtubule polymerization. This is why it is problematic. But colchicine people use in research, even in biotechnological research, plant breeding research. Since chromosome segregation is driven by microtubules, colchicine is also used for inducing polyploidy. You can use colchicine to make diploid, haploid to diploid, diploid to tetraploid, like that in plant cell. That is doubling of normal, diploid chromosome can be done by using colchicine. For example, watermelon, tetraploid plant, one having four sets of chromosomes can be produced by using the colchicine. And when the tetraploid plants is bred back or pollinated by a diploid or normal plant, the resulting seed produces a triploid plant, that is basically a mule of the plant kingdom, and is produces seedless watermelon. So colchicine has been used to produce triploid, that is seedless plant. In case of watermelon and many other fruits, if you have seedless, then it is very good, and commercially you can have high value. So colchicine, the structure is like this, and it is produced from the colchicum plants. It has been extracted, and it can inhibit the microtubule polymerization, and a very good tool for doubling the chromosome number. That means you can make the tetraploid, and then finally by tetraploid and diploid also produced by the use of colchicine. There is another class of alkaloids called indole type alkaloid, aromatic heterocycle organic compound indole. So indole type alkaloids are also present in nature. For example, staurosporin. I also worked on this compound, staurosporin, very interesting compound. This is the structure, is a natural product originally isolated in 1977 from the bacterium Streptomyces staurosporias. It was first of over 50 alkaloids to be isolated with this type of bis indole chemical structure. This is called bis indole. Here you see two indole ring, combines, and it is called bis indole, and this part is sugar. The chemical structure of staurosporine was elucidated by X-ray analysis because it is very complex. So X-ray is also used to elucidate the chemical structure of chemical compounds, secondary metabolites. You know, X-ray has been used to understand the double helical structure of DNA, uh, and it was one of the greatest, molecular work, um, uh, which was finally, uh, you know, uh, deduced the DNA double helical structure, uh, was helped by X-ray analysis. Uh, Staurosporine was discovered to have biological activities, wide range of biological activities, antifungal to antihypertensive. The interest in these activities regulated in a large investigation by the chemist and biologist because this compound has anti-cancer also principle. But the problem is this cancer is a broad spectrum kinase inhibitor, no, protein kinase inhibitors. It can inhibit the protein kinase A, B, C, and it prevent ATP binding to the kinase. This is achieved through the stronger affinity of the staurosporine to the ATP binding site on the kinase. And staurosporine is a prototypical ATP competitive kinase inhibitor in that it binds to many kinases with high affinity, though with little selectivity. This is the real problem of using staurosporine as a medicine because it has no selectivity to specific kinase enzyme. It inhibits protein kinase A, B, C. In my research, I found that it inhibits protein kinase C as well as stop the motility of juice spore. Juice spore motility is crucial for the infection of the plant because juice spore, I am talking about peronosporomycetes juice spore, which are a serious problem in agriculture as phytopathogen. Peronosporomycetes, perhaps you know that Plasmopara, Aphanomyces, Phytophthora, all these plant pathogens produce juice spore, motile juice spore to infect the plant. So if you can stop the motility of juice spore by using staurosporine, then you can control the disease. So I found this compound in a Streptomyces species as a inhibitor or as an inhibitor of juice spore motility. This is no need to study in detail. Yes, this is the last, almost the last slide. The overview of alkaloid biosynthesis pathway. How alkaloids, different kinds of alkaloids, are biosynthesized in the plant. You see L-asparagine, L-ornithine, leucine, and tyrosine, phenylalanine. They are the starting point, and different kind of alkaloids, for example, nicotine, pyrrolidine alkaloids, cholestergens, tropane alkaloids, phenylalanine alkaloid, phenylamine alkaloids, benzyl isoquinoline alkaloids. So these are the overview, most simplified overview of the biosynthesis of alkaloids. There are several other alkaloids here. You see tryptophan and here pyruvate and CAP involved in biosynthesis of some alkaloids. Acetyl CoA mevalonic acid pathway also involved in biosynthesis of glycoalkaloid. Glycoalkaloid means alkaloids containing the sugar. Glyco means sugar. Sugar means glucose, fructose, mannose, and any arabinose, any kinds of sugar if attached to the structure of alkaloid is called glycoalkaloids. So if you are asked what are glycoalkaloids, you should define. So this overview of the biosynthetic pathway you need to remember in detail. So thank you very much for your attention. For dividing cells, this is why in other cells they are not so big problem. This is the good point, in case of cancer treatment. Cancer treatment, it is necessary to be specific. They are specific. They have a high level of specificity to the cancer cell.
Okay, sir. Thank you.
But obviously, the dose is very critical. So, if overdose is used or higher dose is used, even normal cell can be affected. Thank you. And any other? Do anyone have any other question?
Assalamu alaikum, sir.
Walaikum salam, Muaz.
Sir, Muaz here, sir.
Okay.
Sir, I have a question about quinine. Sir, hydroxychloroquine and chloroquine and quinine. These are the are these analogous to each other,
Yes,
or the
analogous. They are analogous, structurally slight difference, like vincristine and vinblastine. They are analogous, but hydroxyquinine, quinone as well as quinoline, both are active principle are same.
sir? Sir, chloroquine and hydroxychloroquine are synthetic, sir. I don't know about them.
Hmm?
Sir,
Hydroxychloroquine and chloroquine.
sir, are they synthetic or organic, sir?
Chloroquine and hydroxychloroquine both are synthetic, but they are they are synthesized based on the quinine, natural quinone alkaloids, because when quinone alkaloids showed activity against malarial parasites, then scientists prepared lots of analogs, isomer of the natural quinone discovered from the cinchona plant, and they found that chloroquine and hydroxychloroquine are more effective, more powerful than the natural analogs, and then commercialize these two products as anti-malarial drug. And in drug discovery, this is like that. The lead compound always a natural compound, and later scientists do some synthetic play and prepare lots of different structural diversity, structural variations, and they do the bioassay which one is the highest active compound. And chloroquine and hydroxychloroquine both are synthetic compound, and they are now a topic of discussion in treating the COVID-19.
Thank you, sir.
Yes, welcome. Any question?
Assalamu alaikum, sir. Assalamu
Walaikum
alaikum, sir. I'm
salam.
Saiful Islam.
Saiful, please.
Sir, in potato and in tomato, sometimes we can see that a parts of a tomato or a potato has a greenish part. Is this problematic for our body or
Good.
harmful for our body?
Yes, it is a good question. Not tomato. Tomato, when tomato is green, that is unripe, then tomato is green and it contains tomatine. Tomatine is very good for our health, skin disease, as well as it has anti-cancer principle. But too much if you take overdose of this type of compound is problematic and harmful. But in case of potato, sunburned potato, greenish, a greenish part in potato. If you buy potato, never buy those, because sunburned part contains, alkaloids, glycoalkaloids, and it is very, very toxic for human. If you take too much, it can even kill the human, and it is a neurotoxin. So be careful about when you take the potato, green part should be avoided. Thank you.
Thank you, sir.
Welcome. Any more question?
Sir, Assalamualaikum, sir. Sir, Sabrina.
Sabrina, please.
Sir, so what is biosequestration?
Sequestration and biosequestration synonymous. Sequestration means many plants produce different kind of toxin, for example pyrrolizidine toxin, and these compounds sometimes used by other organisms of their own protection. Plants produce secondary metabolites or toxic substances for their own protection. When these metabolites are being used for the protection of the herbivore, like insect, I told you the butterfly wings color as well as caterpillars color patterns in the caterpillar, they are basically obtained from the plants they were eaten. So it is called sequestration. That means somehow obtained from the herbs and separate the compounds and deposited in the skin or the how to say wings of the insect or outer side of the insect to warn the predatory bird so that bird or frog through visual observation they can understand the presence of toxin in the insect and do not dare to eat them. And this is very interesting natural mechanism. One organism produces a toxin for its own protection, is hijacked or used by other organisms for its own protection without synthesizing in their cell. Thank you.
Thank you, sir.
Anyone? So today,
Sir,
yes, please. Go on.
sir, I have another one question.
Please.
Sir, Nadia here.
Yes, please.
Sir, can we insert the gene responsible for vinblastine and vincristine production in bacteria and synthesis vinblastine and vincristine largely in industrially, and if this technology is now used by the industry for the production of vinblastine and vincristine?
This is very interesting question and relevant question from a biotechnology student. Yes, vincristine and vinblastine, the biosynthetic pathway has been known. And once you know the biosynthetic pathway, once you know all the genes responsible for this biosynthetic pathway, once you clone those genes, then you can transfer the gene to any other plants. But commercially, till now, no microorganisms or plants or other organisms have been engineered for large-scale production of vincristine and vinblastine. But scientists are trying, different industries are trying to do that. And in case of natural product, this is very interesting approach. Once you know the biosynthetic pathway, and if it is simple, then you can engineer a suitable plant or suitable microorganism, and you can by using bioreactor or cell culture, you can produce large scale of such a complex structure. But in case of vincristine and vinblastine, still not yet done. Some work has been done, but commercially, you may theoretically it can be done, or even practically you can show that genetic engineering is done, but commercially the amount is most important. So in case of vincristine and vinblastine, so far no microorganisms or bioreactor process has yet to be developed, so not yet been done. But please check the literature, because recently I didn't check in the literature. If there is any, please share it in the next class. This is very important question. Thank you.
Thank you so much, sir.
Welcome. Today we have 24 participants. That means participant number increased. We want to see 28 participants. I am not sure we can reach to the target or not. But I understood the internet access from the student side is really a big challenge. Any more question?
স্যার আসসালামু আলাইকুম।
Walaikum salam.
স্যার জিনান, নাইমাতুল জিনান স্যার।
Yes, Naimatul Jina.
Sir, as we know that chloroquine and hydrochloroquine is today able to reduce these side effects in a minimal level.
You know, the COVID-19 is a new disease, and it was first noticed in December last year. And this virus is a new virus, and we know very little about its biology as well as other behaviors. So chloroquine and dihydroxychloroquine, these two compounds have been tested in human physiology. Overdose of any of these compound is very dangerous even for the healthy human. But in case of COVID-19, the patient suffers from very low immunity and several organs failure and other problems. In that case, chloroquine and hydroxychloroquine both are found toxic for the human. And your question is whether the reduction of the toxicity can be possible, but it can be possible, but it is not sure that how these two compounds act on COVID-19 SARS-CoV-2 virus. If toxicity is reduced, and then if they do not work against the virus, then no meaning. But lots of scientists are now working. Only 5 months work on COVID-19. People are targeting how to save the patient, not focusing the mechanism and basic works, but lots of basic works are necessary to do and find the a genuine antiviral drug. But virus is a particle of COVID-19 is a linear RNA covered with different types of proteins, protein-coated RNA. In that case, no receptor, no target sites, not like Plasmodium falciparum, because virus is acellular. So until and unless we do not know the mode of action, you cannot predict that whether toxicity can be reduced or not. But this is interesting subject for research. Thank you.
Yes, sir. Thank you.
Any other question? No? If no question, then I have something to share with you. If I show you quiz question through the slide uh, and multiple choice question, and if there is A, B, C, D answer, And if I ask you to write the correct answer, whether A, B, C, or D, if 10 questions, and if you take a small piece of paper and write the answer, and or and then send me through Facebook Messenger. Is it possible instantly to do that type of quiz? Or if you write in your
Sir,
messenger, seeing the slide A or C or D, question 1A, question 2B or C, and if you send me through Facebook Messenger, I can take the quiz exam very easily, and it needs
sir,
to be prompt. Yes,
sir, yes, sir, Muad, sir.
yes.
Sir, in Zoom, sir, here is a option of sending SMS privately, sir.
Yes,
If
you can send me
everyone,
privately, obviously.
that would be useful, sir.
Yes, you can send me through a message while you are using the Zoom. So you open the message box, chat box, and write one A or B or C or D like that. And when the last slide is over, then you push the button send, so that I can get all of your answer at a time. Is it possible? Can we try in our next class?
Yes,
Do
sir.
anyone have any other thought? How can we take the quiz?
Sir, I have something. Sir, I'm Jahid. I have
Yes.
something to say about this. Sir, if everyone agrees, sir, is it possible to take the classes from Monday to Wednesday?
This is another subject. You know what you can do? You can discuss in Facebook group.
Sir, we have discussed it, so that's why.
What do you mean? Sunday?
Sir, so Monday to Wednesday.
No class in Sunday.
Yes, sir. No class in Sundays.
You mean
From Monday to
Monday,
Wednesday, all the.
Monday, Tuesday, Wednesday?
Yes, sir.
What is the benefit of it?
Sir, other classes, sir, if
This week, Tuesday, I have a meeting. I have a seminar talk.
okay, sir.
Any other question? So how about the quiz exam? The proposal I have given to you. What is your opinion? I want to know.
Sir, I think Zoom chat box will be useful, sir. We can use it.
Zoom chat.
Sir.
Sir, if you do a
Yes,
demo
sir.
quiz, then it will be.
What does it mean? Demo means?
So I mean, as a trial to see if it works out or not.
It should be what is wrong, because you can send me, even if you open your Yahoo Messenger and just write, see the slide and write 1A or 1C or 1D, 2A, like that. So once the last slide is over, then you push the send. Then I will get answer of all of you. It should be okay, and there is no need demo, because what's wrong if you have internet? The only problem is if you have no internet, or if you have electricity failure. Otherwise there should not be a problem. Let
Yes,
us
sir.
let us try in our next class.
Sir,
Okay, sir.
Yes,
sir, I
yes,
have a question.
yes,
Sir,
please.
is there will be any time limitation?
Yes, of course. Time limitation means every slide you will get 1 minute time maximum.
Sir, sometimes some slides miss through, as I have no electricity
I
in my
understood.
house from
I
the
understood.
morning.
Yes, I understood. I understood. Let us try and see what happens. You know, lots of things
Okay,
may
sir.
happen.
Okay, sir. Okay, sir.
Any other? If no more question, I would like to thank you for attending the class, and we are successfully moving on, and 24 students attended in this class. So I am expecting more students in the next class. Thank you very much. Stay safe and healthy.